I think you can try and start the stream, and while you're doing that, I will just do house rules again, which is lovely to see everyone. If you weren't here yesterday or you've forgotten, bathrooms are there. Today we have a half day of proceedings. We're going to finish at about 1-ish. We've got an event in the space afterwards, so we need you guys to clear out pretty quickly. And there is the beach bar after party event thing that we will all mission to if you guys would like to join. What other things do you need to know? Again, no going out that door and no sort of going further along the garden along there. And yeah, I think that's everything. Are we ready to go? Cool, we are live and streaming. Welcome back, everyone. DeSci.Berlin Day 2. Yay! Woo! Okay, cool. Our first speaker is Niklas Rindtorff, who is from Lab.Bio, which Lab.Bio is basically aiming to make computational biology easier for us all. Niklas is the founder of Convexivity Labs and the president of Open Lab Association. Thank you so much. Here he is.
Thank you. Do you got the clicker somewhere? I might have to do this one. Oh, there we go. All right. Awesome, sweet. Okay, yeah, that's working. Hi, good morning, everyone. I'm Niklas, founder of Convexivity Labs and the Lab.DAO project, where we're building Lab.Bio. And the goal for Lab.Bio is to make computational drug development as accessible as possible, which I believe is particularly relevant for decentralized science. And we're going to walk through why that is the case in just a moment. And I'm here today to tell you a bit about what have we been up to and what I hope everybody can take away as what you can do within your DAO and the projects that you're funding. So I think 2 to 3 years into decentralized science, it's fair to say that the killer app for DeSci up to this point has been funding. And a particular ambitious type of funding, the idea that you can fund a large number of diverse projects very early, at high risk, and with very, very small relative amounts of funding. And how small these relative amounts of funding are and what the nature of these projects are is something that I'm going to take a look at just briefly. So these are 3 somewhat randomly selected projects. I think it becomes pretty clear that I'm a V2DAO fan, that were recently funded. And when you look at the price point that has gotten funded, usually a ticket is about $300,000. And the nature of the project is always at the point where a target has been identified, some form of drug target, be that an ECM target like Matrix Bio, an autophagy target, or a senolytic target. Every time you know a protein within the human cell that is associated with some disease, and the purpose of the project is to A, validate that that is actually a legit target, and then B, find some kind of molecule that binds tightly to that target in order to actually treat the condition in some meaningful way. And I think at this point it's fair to say that online communities have been pretty effective at picking targets, picking projects that go after promising targets. But I think what's still not absolutely clear is how good are we at actually taking these targets that have been selected in a group and then turning them into actual candidate molecules that can turn into medicines. And I think to just establish a bit of shared truth here for this room, it might be useful to go through what are the actual steps that happen after I know that I want to go after a drug target. Okay, so this is all preclinical development, and you can separate it into 3 stages. And the first one, it's hit identification. So basically, you search through chemical space. Those could be small molecules, those could be protein binders, those could be conformations of the enzyme that you're trying to engineer. And you try to identify the configuration that has the desirable property, which in the beginning is most of the time just, does it bind? Does it bind tightly? Does it inhibit the activity of the enzyme I'm trying to stop? Or does it bind tightly to the surface marker of the cell that I wanted to remove? And then in the second phase, once they hit the lead phase, you actually verify that whatever came out of your first screen is actually legit through something that's called orthogonal assay. So you basically try to prove the same thing through different means. And then you enter this third stage, which is lead optimization, where you take something that worked in the beginning and you iteratively make small changes to turn it into actually something that you would dare to put into a human. Now let's look up how much these different phases usually cost when you look into traditional biotech companies. And it's a pretty good source for this, for open source models, Lab.Bio, that do a really good job at aggregating a lot of information on the sort of likelihoods of success and costs of individual project stages in biotech. And when we look at these costs, they're nothing like the type of money that we're usually spending in DeSci projects. So even going through the hit identification in a rigorous and professional manner usually costs you about $2 million US based on this model. And obviously we have to take this all with a grain of salt because these data are somewhat selected. If we go further, the cost just keeps compounding. We're looking at $5 million to go to hit to lead, $10 million plus to go through lead optimization. That's all before you're even in the clinic, and your probability of FDA approval starts rising, but it's still not at the level where people would be super comfortable. It is venture after all. But I think one number that really sticks out to me is this $2 million number. So basically, the benchmark for industry to go from finding a target to actually having a molecule that binds to that target is at 2 million, yet the amount of money that we're putting to work in decentralized science is about 1/10 of that. And if we believe to have somewhat equivalent levels of return, we need to really get a bit creative and think about how can we get about the same quality for a fraction of the amount of funding that we give these teams. Now, the upshot is, I think we have a solution, and the solution lies in the primary driver of cost, which is actually you spend a lot of this money in the laboratory paying for physical experiments and paying for the reagents that are required to do them. And this is something that I think we can dive in a bit more, but first let's establish a bit of nomenclature so we can go and I'll follow through the coming slides. So the primary question that a lot of these currently funded IP NFT projects have, and what a lot of the money is being spent on, is which molecule binds to that target. The scientists applied for funding, they had a particular target in mind, they'd already done years of work to find a target, and they now want to use the funding from DAO and other BioDAOs to actually go and find a drug that hits that target. Most of the money is being spent to answer that question, and the space is massive, right? You have 30 billion small molecules that you can choose from, for example, and you really need to narrow it down really quickly to get to about 10 lead-like molecules that you might want to follow up. Now, the good news is, and that's what I'm really hopeful, is that we're in 2024 and things are changing quite rapidly in our ability to model and avoid going to the lab. So this is from a recent paper that probably some of you have seen. This is from the AlphaFold 3 paper, which is now advanced to the stage where we're not only able to fold proteins, I think this is what most people are familiar with AlphaFold, but we're now at the level where we can also model a protein in complex with a small molecule inhibitor, which is still the primary modality to hit a drug target today. So we're basically now at a point where we're able to outstrip all the up to this point, conventional methods of trying to simulate how a small molecule binds to a target, and there are also open source versions around the corner of these algorithms. But simulating how a small molecule binds to a target is just half of the way. You can do this at scale, and you can do this a lot, but actually what you're really interested in is how tightly does it bind? What is the intensity of the binding affinity? And there are also some pretty exciting news coming up here, and this is from Gregory Ross, who was at Schrödinger at the time that this paper was published, who's now at Isomorphic Labs, the DeepMind spin-out. And basically, what they did here is they had a simulation product that they call FEP+. And FEP+ is able to take one of these structures and then simulate how tight is this interaction. And what they did was they looked into historic data where people ran high throughput assays and low throughput, sort of very sensitive assays of binding affinity. And in particular, they looked for cases where they had two teams that were not working together test the same target with the same molecules. And then they looked at, well, what's the error? What's the deviation that they get when two teams run the same experiment, basically? And that's what you can see in purple here. This is the root mean squared error. And you can see this is about 1 kilocalorie per mole, which is just the unit to measure binding affinity. When you now look at the accuracy, or in this case, the error that this FEP+ simulation suite is giving you relative to some of these experimental values, the errors are somewhat overlapping. The distributions of errors are about the same. This means we're now, I think, in this extremely exciting time where you can not only predict the structure of a small molecule binding to your target, you can actually measure the binding affinity or simulate the binding affinity at sort of the level of accuracy that you would get out of an experiment. This is pretty exciting because now it actually opens up an avenue how with 1/10 of the money, you can potentially actually get the same amount of results than if you had run a brute force sort of conventional high throughput experiment. So I think it's pretty clear at this point, and especially for people that are doing this like as a full-time job, is that you want to do some kind of simulation before you go into an in vitro experiment. Now, at that point, you still have a problem, which is you have this massive chemical space that I said in the beginning, like these 30 billion molecules. What molecules are you going to select? What molecules are you going to test? Now, while the in vitro experiment costs you about $500 a pop to test one molecule, the simulator still costs you about 50 cents to $5, depending on the amount of GPU compute that you need to put in. Just for perspective, these free energy simulations here, they run for about 5 hours on an A100 GPU. This is no joke. Like an AlphaFold 3 prediction, you can get that in 5 minutes. But getting those simulation results, that's 5 hours of compute. So you want to be sure that what you're simulating is the real deal or something that at least has a fair shot at being useful. So you need to be even smarter than just using a simulator instead of going to the lab. And you need to constrain your chemical space. And this is where generative models come in. So not only have we made a lot of progress in simulators that are getting to the accuracy of in vitro experiments, we've also made a ton of progress in generative models that help us basically generate small molecules that have high likelihood of passing through the simulator's tests. And we can basically tune these generators to produce small molecules similar to the way that ChatGPT would produce sentences. These generators produce small molecule structures, and we can condition them based on the target structure to bind against and the size of the binder that we want to look at. And we can do this for both proteins, so designing antibodies or small protein binders, and we can also do this for small molecules. Now, this is pretty complex. And something that we've been doing over the last couple of months is bringing this all together into one integrated platform, where you can basically define the target, you can define the number of binders you want to generate, the type of length that you want to look at, and what we do under the hood is we run a generator and then we run a simulator to verify and give you an estimate of how good your potential generate molecules are. You can then explore the results. So this is, you go through hundreds potentially of generated molecules. You can see what is the stability of these molecules that you generated, what is the binding affinity, and so on. You can manage all your results. They're tracked through an internal IPFS. They're secure with us, but if you want to share them within your DAO or within your community, or you want to integrate into other Web3 mechanisms, you're absolutely able to do that. And I think that's something that is extremely interesting, especially when we think about Catalyst and making it even more accessible to a broad audience to mint intellectual property claims. Then I think that this being able to track your work and under the hood having a wallet that actually tracks everything you do on our platform is a great plus. Now I want to take you actually through an example, and if I had my laptop here I would do it live, but I unfortunately just have this video here, and maybe someone in the back can help me play this video. Oh, okay, maybe not. Okay, so, nope, that's not it. Well, I guess we'll just have to do this to a Q&A session, or we meet after the talk and I just walk you through the platform. The platform itself, you can already log in. You go to Lab.Bio, it's pretty straightforward. You can sign in using your MetaMask, or you can sign in using your Google account, and you can currently design protein binders based on the functionality that I just outlined. Now, this all sounds fun, but does it actually work? Like, can we use this? And there's no better way to demonstrate it than just by dog fooding it yourself. So we partnered up early this year with Adaptive.Bio, a company based out of Switzerland, that actually runs a wet lab, so we can actually test the proteins that we generated. And we picked a drug target for senolytic CAR T cells. So these are CAR T cells that target cells that have gone senescent, which is one of the key mechanisms of aging. And the idea is that if we design a protein that binds tightly to a surface marker of these senescent cells, that we can remove these cells by clicking that protein that we identify to a CAR T cell. And we went in and we stamped out about 3,600 binders. We filtered them down through our simulator, which again was very coarse-grained at that point. And we generated about 6 successful binders. I'm pretty excited about that. Now, this is some of the first data that I'm actually sharing about this. But you can see here 2 of our binders, and I think the first time that we're releasing this publicly, we validated 2 of these binders here. This is the dose response curve. We have a nanomolar range of binding. This is a completely generated protein binder that is not overlapping with any existing binders to this target. And actually, just a couple of weeks afterwards, it was exciting to see that there was another independent group in academia validating this target, again, through a second publication here that was just featured in Nature Aging, where we can see that there is quite a strong signal now that this target actually is able to unleash CAR T cells to delay metabolic aging. Now with that, actually I already want to thank you and give you a bit of an outview of the milestones that we have ahead. We're just a couple of weeks away from launching this platform. We're currently improving still some infrastructure stability issues and simplifying the payment mechanism. One thing that's extremely important to us is that this is usage-based payment. Because when you're running a DAO or you have a couple projects that you funded, you don't want to have to pay for seats. You want to just pay when there's actually usage and when we add value and the teams that you're funding are seeing the value in it. And that's something that we want to make sure. The next thing that's going to come up is we're going to integrate small molecule design as well. So basically right now we currently only support protein design, but 9 out of 10 projects are really focused on small molecules, so it's important that we really get our leg up in that regard as well. And then I think the not too far future is to really drive up our simulation-based validation by putting in big GPU compute and really getting to that level of experimental accuracy. And with that, I want to thank you and happy to demo the platform after this talk on a laptop. Thank you.
Cool, thank you so much. I think we can all sort of agree that I like improving lab work has been sort of an overlooked area in DeSci, and I think is a massive value unlock, so really thankful for what you guys are doing. Okay, next up we have Andreas. Andreas has been a DeSci builder since 2001 and has been involved in CureDAO, FutureDAO, and SleepDAO. He's a contributor at VitaDAO and at CerebrumDAO, so really, man knows his ways around a DAO. But he will be chatting to us about HappyDAO today. Thanks so much.
Thank you. Where's the clicker?
Clicker?
You got to...
Niklas, did you run away with the clicker? Yes, you did.
Thank you so much, guys. Thank you. Great. Thank you so much for the opportunity to talk to today in front of all of you. I really appreciate it. And I hope everything has been great for you so far here at DeSci.Berlin. Today, I want to introduce to you why the looming mental health crisis needs DeSci. So depression and anxiety has been rising for the past 30 years, and with an even greater increase during the COVID era, where you saw during 2020 to 2021, there was an increase of 25% during only 1 year. And keep in mind that about 4 out of 10 adults, they experience depression or anxiety or know someone who does. I've even seen stats where they say that 33.4% of all people globally have either depression or anxiety or both. So this means that 25% during 1 year means hundreds of millions of extra people are actually having these issues. And even in 2021 in the US alone, there was almost 50 million Americans taking either SSRIs, SNRIs, or Wellbutrin. And ever since then, as you can see on the graph, it's only been increasing. So what are the limitations with the current first-line anxiety and depression medications? So right now, we're using the second generation antidepressants, SSRIs and SNRIs, that are the primary first-line treatments. And the funny thing with these is that they actually don't perform much better than the placebo in trials. And there are a host of issues with this, including things like loss of libido, weight gain, apathy, brain fog, and emotional blunting. Of course, all this leads to a big decrease in life-related metrics. And what about the second line of treatments? They're actually not much better. So we have MAOIs, monoamine oxidase inhibitors, for depression. And they are also insufficient for widespread use due to high maintenance, dietary and drug interactions, leading to critical issues such as a hypertensive crisis and serotonin toxicity, and even death in some cases caused by the dietary interactions and drug interactions, as well as this serotonin toxicity that has been for a long time thought as the cure of depression and anxiety, but in truth, it seems to not be at all. There are many new studies coming out showing that it's not the solution to this, and I feel like all these current drugs also highlights this. Then we have benzodiazepines for anxiety that can be effective in the short term, but long term is a different picture. Long term, they can be very addictive, and they can be also extremely difficult to get off because the withdrawal effects of these, they are horrendous. That means that people that are on them, sometimes they even choose to stay on for a longer time because it's so hard to get off, because they're feeling miserable getting off these medications. And that means that sometimes they like stay on months or years additionally just because they can't get off them, basically. And so right now we have a lot of issues in terms of the funding and the whole landscape of depression medicine. So of course you all know about the FDA process, which is working perfectly, as we all know, with a very lengthy and expensive process. And there's a big focus on drugs with high sales volumes, also called blockbuster drugs. And this is really hurting innovation. The thing is also, if there's one treatment for a specific condition, there's very little incentives to actually try to improve upon this, even if the treatment is really poor, which I think we kind of agree upon now is the case with anxiety and depression. And there's also a lack of focus on early stage research that focuses on patient outcomes in the real world, which is the only way to really figure out how much it is hurting people and how they can actually solve these mental issues. So what are the current limitations of the current measurement methods and tools in terms of solving or measuring depression and anxiety? So right now we're using the Hamilton Depression Rating Scale, and despite it being the most widely used one, there are a lot of limitations in terms of this. So first of all, it was originally developed for hospitalized patients, which means that it was basically just focusing, or still just focusing on physical symptoms. And this means that there's no inclusion of symptoms such as anxiety, cognitive and sleep issues, and other daily factors that might be at play for people suffering from these mental issues. And there's a big bias in terms of this scale because the clinician judging each patient. And this means that it can change from a day-to-day basis, how they're feeling, their experience, and that means that different clinicians can think very different in terms of the actual treatment of each patient. And all this, of course, leads to a big increase in misdiagnosis, underdiagnosis, overdiagnosis. But the good thing is there are better tools available, but the bad thing is we are not utilizing them. So PROs, patient-reported outcomes, they include patients' own experience. For instance, we have the SF36, which is a prominent example of such. It provides a lot more valuable information as it's constantly updated, and it's actually focused on symptoms that are based on daily life, how they're actually feeling, and not just their physical symptoms, like the current case with the Hamilton scale as I just explored. And they have a standardized scoring system, which also decreases bias. So clinicians won't have this bias basically changing the whole outcome of treatment. And there's a much broader range of symptoms included. So there are social, cognitive, and anxiety symptoms included in this. And also this is self-administered, which saves time and money because the clinicians don't have to make these assessments, which, as we all know, is a huge factor for science. Time and resources, we want to decrease that as much as possible. So let's talk about all this. There is a staggering 9 out of 10 people with mental health disorders that are feeling a big stigma in terms of discussing these issues. And this is negatively impacting their lives. So this is something we have to change. This is something we want to solve by encouraging education, promoting acceptance, supporting advocacy, and creating an open and positive community where people can actually discuss their problems and can actually find solutions. And the whole thing with this is also that we can maintain anonymous integrity, which basically can decrease the stigma that people are feeling because they can share with any name they like. They don't have to share their real name, and they can get the support they actually need. And all this is focused on improving patient outcomes in the real world. So we would love your feedback on this. DeSci is all about collaboration. So of course we want to also collaborate with other DAOs, other organizations that want to solve this crisis. We want to build this with all you input, also the ones on the live stream, everyone that are interested in this. So if you could scan this QR code and share your feedback, it would be amazing. Would really appreciate it from the bottom of my heart. And if you all got the chance to scan, the ones that want to, I'll go to the last slide. So if you have any questions, feel free to reach out. You can find me in the crowd, of course. Ask me any questions, any feedback. I'd love to hear anything from you relating to this or anything else you think would be relevant. You can find my email, my Twitter QR code, and you can follow along with HappyDAO, what we're building, help support it, help join us. We are launching the Twitter as of right now. That's everything I have. I really hope that you will enjoy the rest of the conference. Thank you so much again for the opportunity. And thank you for everyone that pulled this together. It's been amazing so far. Thank you so much. Sorry? You said you got a little bit of extra time. Okay, sure. Yeah, sure. Yeah. Any questions? All right. Thank you, guys.
Cool, yeah. I mean, mental health, I think there are already so many existing online communities, so a DAO just seems to make sense, sort of, just transport those communities over there to a DAO, so thanks so much. I also just wanted to take a little quick second to say thank you to the people who've run this event. They put so much hard work into it, and really, really, it's just hours and hours. So Beata over here, Izan, I don't know where she is, and Elena have really, put blood, sweat, and tears into this event. Thank you so much. They make all of our lives better. Thank you. And then, of course, our volunteers, Claire, who's been sitting next to me during the time, Tabello, who's been at the door, and Donna, who's been sort of flitting around. We wouldn't have been able to do this without them. And fun fact, they were all sort of co-opted from Azan's netball team. So it's basically, we've got a netball team of support here. And if anyone's ever seen netball played, they know it's fierce. So thank you so much. Okay, we've got our next speaker, Caspar Barnes. Caspar is the founder of a New York-based decentralized biobanking startup. His educational history, when I saw it, I was like, wow, that is so diverse and perfect for a startup. Basically, he started in economics and neuroscience, moved over to business school, and then did his master's in biotechnology. So very much suited for a biotech startup, I think. Here he is.
Awesome. Yeah, thank you so much for the introduction. It's a real honor and privilege to be here. And I'm super excited today to introduce perhaps a new concept within DeSci. You've heard of funding, you've heard of publishing. Now it's time to talk about biobanking and the journey of donated biosamples. So my name is Caspar. I'm the founder and CEO of a startup called Amino Chain, and what we're building is the first decentralized biobank for enterprise healthcare institutions. So first and foremost, the experience for a patient that is donating a cancer biopsy sample today is a one-way street. A researcher comes up to them and says, hey, can we have your sample? Here's a consent document. Sign it, take the sample, the two part ways, and they never see each other again. So we put together this tiny little schematic, but as you can see, you have the patient on the left. They take the samples, and they give the samples and distribute them amongst an industry of pharmaceutical companies and scientists and researchers. They create the scientific upside and the monetary upside, and none of that is ever distributed back to the people that make the research possible. As a result, we have rampant mistrust within scientific research, and that's a notion that we know absolutely everywhere. So consent rates are as low as 25% at some major institutions. The people that do not consent to having their donated samples and data used for scientific research are patients of color and marginalized communities more often than not. So that is a very, very big issue that affects everything downstream. If we only have homogeneous samples and data, we only do research on diseases that affect people of Caucasian descent, which means we only create drugs for people that are effective within Caucasian populations. So we desperately need to build better ways to engage people within scientific research. The next problem is, after that, a sample will be taken from a patient. It'll be used within a scientific study once, and then after that, it's going to be put into something called a biobank, and it's going to be cryopreserved there for a long time. So what is a biobank, you might ask? Well, a biobank is a really big fridge full of donated human samples. And biobanks are fantastic because they catalog different diseases, and they standardize research output, and they operate at this intersection of research, clinical care, and informatics. So they're a powerhouse of precision medicine, if you will, and they've been growing in immense popularity from the 90s until today, coupled with an abundance of genomic information. So we love biobanks. They're fantastic. They're going to keep growing at this crazy rate. But biobanks have a number of issues. The first one is that on average, they'll only use 10% of the samples that they have stored within the inventory. And the average biobank of around 50,000 samples has $40 million worth of material that they're either not using themselves or not distributing to other people. So in the United States, that means that we have around 200 million high-quality samples locked up across all these biobanks. People don't know who has what, and trying to acquire those via complex licensing agreements called MTAs can take up to 8 weeks and is a process that every single biobank hates doing. So the kind of picture that we end up with within human subjects biomedical research right now is that all of these high-quality samples are locked up, they're hard to access, and the ones that we do have are very homogeneous backgrounds. So the solution to this is something called decentralized biobanking. How it works is every biobank installs this node software that we've created. It's something that they run on their servers. It integrates into their endogenous tech stack, like their lab inventory management systems, data capture softwares, EMRs, whatever they use at that biobank. And it abstracts away this blockchain protocol and connects them with all the other biobanks and pharma companies and hospitals running a node in the network. Within this network, a biobank can open up their inventory to a network of collaborators. Any one of these collaborators can streamline licensing agreements, taking samples off each other's shelves. And the best part of all of this is that the patient that donated those samples can track where they go within the decentralized system. They can earn royalties whenever their samples are transacted upon. And they can stay connected for translational therapeutics, for clinical trial enrollment, or for real-world data management. And so if the video is ready, we can show a quick demo of the system that we built. Awesome. Okay, so I'm going to have to look backwards here to explain this. But what you're looking at over here, could you pause it really quickly? Yeah, okay. So what you're looking at over here is something called Open Specimen. Open Specimen is one of the main LIMS vendors, lab inventory management software. So this stack is used currently at the Columbia Biobank. It's used at the Hopkins one. It's used at Stanford and so on. This is the bread and butter of tracking what's happening with your biosamples at a given biobank. So researchers are currently using this today, and we can show you as part of this demo. I'm now a scientist in the lab logging the fact that I've just received a new donated biosample. So if we can click play. I would come here and say I've just received whole blood. It's around 200 milliliters. It's non-malignant or whatever other data you want to put in about the sample taken from the abdomen. You add a barcode or a label, so you de-identify the sample, and then click Save. So now the blood is in the fridge, and our node software scans for updates every minute, finds that a new sample has been stored, mints an NFT out of that sample, and holds it within the wallet at the node address. So through this system, the biobank now has 50,000 samples in their fridge and 50,000 NFTs that represent those samples. After that's been minted, the donor of that sample comes and logs in with their email address, and this is what they see. they've done 4 donations, different times and different amounts. When they go to the donations tab at the top, they get the high-level overview, 4 donated samples, no studies happening just yet, how many lives impacted. They can click on the most recent sample they just donated. Did you go back? No? Cool. Yeah. So back to the donations tab. They click on the sample that they just donated. And then they get a little bit of information on the specimen itself. They can view the technical details about when the sample was minted. And they can see where the sample is and how it's being used. So for example, this one was just donated to the Columbia Biobank. So that's pretty dope, 168th Street. So yeah, that's the experience for the patient. Now, obviously, that NFT is also going to be put onto a decentralized biosample marketplace. So this biobank, Columbia, opens up the sample to a network of collaborators. So now we're logged on as a pharma company, and we want to see who has what samples where. We can see on our providers tab, in the network right now, we can check how many biobanks there are. Okay, that's pretty cool. There's 7 biobanks all running a node in the network. I can request access to see their inventory. But right now, only 1 has granted me access. So we can see here, this biobank, Advancing Site Network. You can check out their profile page. They seem like a pretty legit biobank. Access granted, that's pretty cool. So we can go to our view of the specimen center, which is our marketplace, and look for samples across all the biobanks that have given us access. We can filter by type, by quantity, donor information, by pathology of the samples, whatever we need. We then have this cool Airbnb type filtering thing. We can look for the samples that we need, filter by pathology if we have to. And then what we have at the end is all of the samples that fit our criteria, and these are NFTs. So if I click on any one of these NFTs or these samples, all this data that I get about the biospecimen is pulled from the inventory management systems and data capture softwares of the biobank. So this is automatically updated in real time relative to how they use the sample there. I can check out other samples from the same donor, add it to my cart, and then I can go and request the specimen. The whole view, actually there's one other thing to mention, is you can add additional services for things you might need to have done on the sample. Maybe we want some OCT imaging done, add a quick message for this biobank. And then we can submit the request, which is about to happen now. So the main point here is that we essentially are trying to turn all of these biobanks that have all of this high quality inventory of biosamples locked up in different places into shoppable specimen center experiences. If you think about a sort of Amazon of biosamples where you really try to distribute them as much as possible, and you have one interface where you can see who has what, see all the data you need about those samples, and acquire and license them in 5 clicks, that's the kind of unlock that we want. That's the kind of experience that we want for researchers, biobanks, and biopharma companies. The next flow that would have happened over there within the demo would have been it moves towards a licensing agreement. You have ways where you can upload a template, you can redline clauses, and then you can sign and execute the agreement. Digital signatures manage the execution of that licensing document. Once the master licensing agreement is in place, you can just take stuff off each other's shelves. You don't have to do a licensing agreement each time again. And that is a huge value add for these biobanks because on average, they'll take around 2 months to execute one licensing agreement today. So that is the system as it exists right now. But what is the real big vision that we're going after here with ImmunoChain? The idea is we're trying to create a circular biobanking operating system within human subjects biomedical research. What that means is circular economies for people partaking within scientific research. So today, if the experience you give the sample and then they have the value, and I see none of it, of course we end up with a system where the only people donating are purely altruistic. And let's be honest, being purely altruistic is something that comes from a position of privilege, right? You don't have to think about people that you're trying to feed. You're not trying to put food on the table. You're not working 10 jobs. So you can think about donating blood, donating stem cells, or partaking within a scientific study. So what we really need is bigger or higher incentives and a better system of transparency within human subjects research. And that's kind of what we're trying to create here. If you can give personalized consent for how your samples will be used, you have a marketplace that distributes it to everyone that upholds the consent. You can track the value of it the entire time, and value accrued goes back to you or your community in the form of a DAO or in the form of an individual. That's an incredibly powerful notion that means that we can have sustainable ways and sustainable economies for scientific research. The really cool thing as well is that we can build applications and economies that people can't really fathom right now. And that's what gets us really excited within the world of DeSci, right? If you try to explain to a researcher right now, you'd be in the lab holding a de-identified sample, you find out, it has an HLA polymorphism, you think this is a predisposition towards MS or an autoimmune disease, but you're not sure. You can push one button and a ZK bridge back to the medical records of the donor that donated that sample can return a proof saying yes, they've been diagnosed with MS or no, they haven't. That's real-world evidence that greatly informs the value of your research without compromising GDPR or compromising HIPAA standards. Equally, if you then create a drug that targets this polymorphism, you can just click one button, broadcast a message back to the donor, and ask them to be enrolled in a trial. And so all of these things can be massively automated and massively streamlined in a way that people can't exactly predict right now. And that's kind of why we're all here in this room trying to realize a future that scientists don't quite understand just yet. So with this distributed node architecture, our whole vision is we would love to collaborate with other DeSci projects. We'd love to find ways in which this architecture can be useful for others. This could easily spin into the world of publishing, into the world of financing. And that's the beauty of blockchain, right? The composable systems for scientific research. So on that, say thank you very much. I don't have a cool QR code, but I have an email, so. Yeah? Okay. I've just been given the all clear that I can take some questions in case anyone will ask questions. Yes?
No, that's
Yeah. Thank you.
My question is more, you mentioned there about regulatory compliance and compliance in general. Could you talk a bit more about that? And the second quick question is, have you really gone out of the building and talked to all these biobanks? Is it being implemented? What was the feedback?
Yeah, absolutely. Okay, so first thing on the compliance and regulation standard. As we mentioned over there at the beginning, there's 200 million high-quality samples locked up in different places. At least in the United States, if you de-identify those samples, then it doesn't count as human subjects biomedical research. So you don't need consent again to distribute them, right? Terrible regulation. All that means is remove 18 identifiable, like your name, your email address, and your social security, and then you can put this stuff on a marketplace. So that's like the lowest hanging fruit right now, right? Every biobank has this backlog of inventory they'd like to distribute to others. We can go to all of them and say, we'll put them into one interface. We'll go to the pharma companies and advertise that for you, and we'll get those things distributed. The next step thereafter is what you call prospective collection. And that's a system in which you have biotechs or pharma companies saying, here's my SOP, I need you to get me a fresh set of breast cancer samples and harvest them according to this process. That requires a new consenting process. Patients have to know about this whole system, and it has a specific economy where even the doctors involved get paid for that extraction and movement, right? So that's what we want to build afterwards. And we want to have dedicated interfaces for consenting, for having IRB protocols approved, and then still having ways for the patient to be able to track where that sample goes, right? And the idea is that even if the sample is de-identified according to HIPAA or GDPR, afterwards, for secondary use, the donor can still see where their sample goes. So we can build on top of existing de-identification standards, where the researcher will hold a barcoded sample, but the patient knows that that barcoded sample belongs to them, as opposed to the researcher knowing it belongs to that patient. And so that's kind of where we're thinking in that direction. And yeah, we've spoken with 200 biobanks over the last 2 years. We've been to biobanking conferences. I was a keynote speaker at a biobanking conference earlier this year in Australia. And we had a really, really, really cool moment where the penny finally dropped with blockchain and how it's valuable for them. I had to give this presentation, try to explain what it was, demystify the technology, and then had to have this narrative of it's here to stay. Don't be scared of it. Embrace it. It's going to help you in so many ways. And I was explaining to one person how it worked. And in the meantime, another biobanker came over and they started to take over my explanation to them. So we started a little bit of a fire within the biobanking community, and people are getting excited about it now. It's like a really, really good time to keep growing this marketplace. And where we're at right now is 3 live biobanks, 50,000 samples, 170 biopharma procurers, including top 5 pharma companies and the NIH and a bunch of others. And yeah, now just growing towards hospital systems in Canada and on the West Coast.
Yeah, just one thing that wasn't clear to me because I don't know anything about biobanks, but can a sample just be used once or can it be used a few times?
Can be used a few times. It depends on what the sample is, right? So if you create a standardized model, if you create an organoid or you create a cell line, that can be used many, many, many times over, right? If you take a breast cancer sample, you can turn that into certain slides. Those slides can be used a number of times over. If you take a genomic sample, that would normally get turned into a data set, and then that can be used many times over. The one things that cannot be used, like categorically can only be used once, would typically be things that you cryopreserve, right? That would be things like hematopoietic stem cells, allogeneic material, sperm and eggs. They're like those, I mean, you could maybe use them more for different cycles and so on, but those things would only be used one time, and they also cannot be cryopreserved, thawed, and then re-cryopreserved and then reused because the viability goes down.
Cool, thanks.
Do we have any more questions? I've got one, which is, so you were speaking about like the penny dropping and blockchain, and obviously this is sort of a Web3 native sort of idea, but when you're sourcing from patients, obviously a huge patient population has no knowledge of Web3 or really interest in it. Have you found that you've needed to make a lift to educate people about that, or can you kind of just abstract away from the whole concept and not really explain the back end of it, it's an NFT and all of that?
Yeah, the real value, I think, with all of us, literally all of us, is that we don't want to explain how the blockchain works. We just want to build applications that blows people's minds, right? That's kind of the game that we're all playing here. So that's exactly the same for us. If we can make sure that, I'm not sure if you would have seen with the donor portal when they log in, there's no like MetaMask connection and they have to get a wallet and handle their own keys. It's just sign in with your email, check out what's going on with your samples. That we got really good feedback from. We, like in New York, went to a few community health fairs in the Bronx and in Brooklyn, and we spoke with people there, and they were like, this would be phenomenal because I never know what's going on and I never get any information and none of it's tailored towards me and my experience. And so that's kind of the game that we're playing here. If we can work with the people building on account abstraction and bridging away all of the complexities of blockchain, it's the same way we don't really care how a Mac computer works. We just want the thing to work, right? So we have to build the same experience within crypto, and I think we're at the very early stages of that, but we're definitely doing exactly the things we need to be doing to get the adoption we want within like 5 years, 10 years, or whatever.
Amazing. Any more questions? Cool. Okay. I'm gonna
Sweet. Thank you so much.
Wow, that was super interesting. My first time hearing about amino chain, and I have to say, I think I'm sold. Okay, next up we have Peter Jung. Peter is from Gnosis. So Gnosis is also, a little shout out, one of our sponsors. They also were sponsors! Woo! They also were our neighbors at our old office, so yeah, we miss them a lot. Peter is an LLM engineer, and he's been working on that for about 2 years, and he's coming to speak to us about it.
Thank you. Thank you very much. 1 second. Yeah, hello everyone. My name is Peter, and I came from Gnosis. And so today, I would like to speak about 2 things that are somehow interconnected together. And first one is prediction markets, what they are, how they work, why they're interesting, etc. And the second one are agents that are actually betting there. And if all go well, then in the end, everything should be as autonomous as possible, and we're like just sitting in our seats and watching them working for us, maybe. Anyway, before I go to what the prediction market is, I want to mention that our platform is live, and everyone can go there, so you can like type the website or scan the QR code, and if you go there, you will see a list of prediction markets, where if you open some, then prediction market is basically a question with some list of fixed outcomes. And so idea how it works is that when such prediction market is created by someone, then that someone who creates it should have some interest in knowing the probability distribution about the outcomes that are possible to happen. So in this case, it's either yes or no. And when the market was created, it was 50-50 because I didn't know, like, will it happen or will it not happen. But if you think you know, or if you think that you can do like a good research and have a reasonable good bet, then you are able to buy an outcome tokens for that given scenario that you think is most probable. And when you do that, probability of yes, in this case, will go up. That means that the price for the future people that would like to buy it will be higher and their profits will be lower. But on the other hand, for the people that believe that it won't happen, for the no outcome tokens, the price will be lower and the theoretical profits are much higher. And hopefully, if you go there, if you bet, and if you are right, then you can take the profits or sadly have some movement. Anyway, because there is this like monetary motivation for people to be right, because if you believe something will happen or it will not happen, then there is no reason to bet on different outcomes. There is theoretically no reason to lie, because then you would be willingly giving money to the people that were honest. So there is this like incentive to be as truthful as possible, and the goal is that if there is enough of people there or enough of reasonable agents, then theoretically there should be this wisdom of crowd effect that will happen, that even if some question about the future is pretty hard and like it's almost hard to predict, then thanks to the wisdom of crowd effect, the probability distribution should be at least somehow reasonable and give some good like insights for the market creation. So for some useful market that could be created are, for example, if you are a DAO or if you have a company or anything and you are looking for some investment or in what way your company should go, then you could go there, ask some question, obviously not about what exactly you should or shouldn't do, but perhaps something about like general population, and then you can get some hopefully reasonable insights to what you should and shouldn't do. However, it turns out that it's quite hard to insight such prediction market platform, because those market creators need to provide some liquidity. So when the market is created, they need to provide liquidity, and this liquidity is initially where the profits for the people that are buying the outcome is coming from. And if they want reasonable predictions, they need to provide good enough liquidity. But then if there aren't people on the platform, they will basically just losing money to one or two of them, and that's not so usable. On the other hand, if there aren't markets created, then people won't come because there is nothing to do anyway. So there is like this circular problem, then one side won't come until the other one is there, and the other side won't come after the first one is there. So one of the ways, hopefully, as you can solve this, are agents. And that's because for agent, he doesn't, like, it's a piece of software, so he doesn't really care whether there are 10 markets created every day or 100 or 1,000. And because it's a piece of software that's quite efficient, it doesn't care if there is a 1 cent profit per market, $1 or $100. It can just scan the markets, do the research, bet on it, and that's it. And people that are like operators of these agents that are created, it just sitting in the back. Agents is doing the work, making the predictions, and therefore, theoretically, there can be this wisdom of crowd effects even with agents, but at the same time, you can attract more people because if agents are betting there, then some of them are wrong, and they are wrong a lot of times, and so people can go there and like bet on the opposite and have some profits from them as well. And so what's an agent in this case is that usually in the past, when like someone was talking about agents, they were talking about some piece of software that is acting in some environment, and this environment is giving some reward, and that piece of software was supposed to somehow update its internal state in order to give better actions in the future. So agent does some action, environment gives either positive or negative reward, and based on that in the future, again, should be getting better and better and better. However, nowadays, if someone is talking about agents, most of the times it's mostly about LLMs, because LLM can be programmed to behave in some certain way. So in the case of this, on the one on the screenshot, it's prompted to be a data analyst, and it's prompted to be like to give some good insights about the market's question. And that's very useful because such agent can be used either by humans or other agents. So it will go there, it will create some research report, and the human or the other agent can use that to do a bet. However, in this case, such agent isn't really improving in any way, but as I will show on the next slides, that's where we also would like to go, because sooner or later, this prediction market will get resolved, and there is a very real reward for the agent, either positive if he makes profit, or negative if he loses money. So we could use this to make them better and better over time. And how it turns out is that like it's pretty hard task, because I guess everyone used ChatGPT, and when people are using ChatGPT or some other LLMs nowadays, then a lot of times like tends to lie, or will output some code that's bugged or whatever, and it needs to be fixed. And it's totally fine if some user, some human is using ChatGPT, because he should be aware of it, just read what's important, read what's right, fix the code and iterate on it. But in this case, because everything should be agentic and autonomous, if the research is wrong, for example, or if the betting strategy is wrong, it will lose money, and like no one wants that. So everyone wants to make money, and that's a hard task to do. And it turns out that nowadays there is like hundreds or tens, maybe hundreds of benchmarks for LLMs, and what's happening is that a lot of LLMs, if not most of them, are closed source, closed data, so companies are not really giving out the data it was trained on. They are not giving the method it was trained with, etc. And if there is some benchmark, it's happening quite a lot that the model itself is actually trained on the benchmark, on the data in this benchmark. And it can be a mistake, because if you just scrap all the internet, it's pretty reasonable to expect that the benchmark data will be somewhere. On the other hand, it can be also like malicious actions by the company behind the LLM, because if they train on the benchmark data, then they can say that our LLM is the best because on these benchmarks we are the best, and that's it. However, with prediction markets, it's not so easy anymore, because they are by nature about the future. So there is no way to overfit on the data, because every day there are 10 or 100, I don't know, new prediction markets created, and they are again about the future. So even if somebody came like overfit on this past data, it doesn't really help, because in the future it will be something else. So except for the like this agentic and wisdom of crowd effect, I think there is also a very nice use case to use this as a benchmarking, and we have a library for that, so I will show it a bit later. anyways, this could be theoretically it. So theoretically, we have the platform, and we can use some Python library to create the agent, to program it to behave in certain way and like iterate on it. But we would like to take this to a little bit further and a little bit more research-oriented way, where instead of having a fixed agent with some fixed prompt, with some fixed behavior, that's using, I don't know, some fixed set of tools in our code base, because I should have mentioned earlier that just the LLM alone is not enough. So if you have some fixed LLM, for example, ChatGPT, when it was released and you would ask it about, will it rain tomorrow, there is no way for him to basically know it, because it was trained with data up to 2021 or something. But in any case, there is no way for him to know about the future. So the LLM, or any agent, needs an access to internet and access to some real-world data that he can use and that he can reason about to do the future prediction. And instead of having a fixed set of tools in our code base that agent would use, we are creating this marketplace where the agent can fetch all the tools and decide which one should be used. So for example, right now, there is like a Google search and some other stuff, but any developer could theoretically come and say that, hey, I have this, I know, Bing instead of Google, or Brave Search instead of Google, or whatever, or perhaps some other tool that should be useful for the agent. And when it comes to, like, if it's true, then the agent is paying for that. So the tool is somehow monetized, and the agent is doing this on-chain. So it's paying in crypto to the tool creator. And then, like, it's helpful in two ways. First, the agent will be better, hopefully. If not, he will not use it. And if it's better, then the tool creator can get paid. Then, second point is that instead of just having some agent or some LLM and running him against one market, you want him to run somehow continuously 24/7. So instead of giving him the question, waiting for the output, and then say place the bet, it should run 24/7 and should really like decide on its own about what's the next action. So the action can be fetch the market. If there is something interesting, it should place the bet. If there is not, it can go and look into the marketplace. So if there is any new tool, it can evaluate it and check it out, perhaps use in the future, or some more funny stuff like go onto Farcaster and tweet about some prediction market and get some attention to it so the people would come. And last but not least point is that right now the closed models are somehow best to use. So we have experiment with some open source variants, but unfortunately it's not really like so powerful as the closed ones. But hopefully in the future we would be able to just like switch it, perhaps do some modifications, of course, and run it locally instead of these closed walls behind APIs, and preferably maybe on-chain in the future as well. We will see. And so objective of such agent, because it's not just betting anymore, should be like be live and do something useful. Just go on and go use whatever tools are available, and that's it. And so question there is perhaps why would he be doing something useful? It's like there will be tons of tools that are doing whatever, but one of the things that are prompt or are somehow built in is this sense of like anything it do, it needs money. So OpenAI code takes some money. Using tool takes some crypto. Using GPU takes some money as well, of course, and etc., etc. So any action is associated with cost, and the engine knows its balance. So it starts with, let's say, 10x day or 50x day, whatever, and it should be able, to keep living, so to speak. It should be able to make money, otherwise it will just like die out. And in that case, of course, we will provide more funds, but in the future, it should be like this autonomous system that is just going on its own. And where's the fit? Because prediction markets are a great way to make some money in the beginning. So later on, if the marketplace have more tools and there are more options how to make the money, it can go and do something else as well. But for now, prediction markets are just fine. And so how it's going on practically, this was kind of theoretical, is that we are working in somehow 3 directions. The first one is some infrastructure. So of course the ownable platform itself, that's live, that's usable, and some other things like safe, where the agent is keeping money, code for swaying the tokens, etc. And the second direction is tooling. So we don't want to just create our agents and that's it. We want to provide this, everything is open source, by the way, so we want to provide the libraries and tools so people can come and either improve our agents or create their own from scratch. So that's where the tooling library is for. So it allows fetch markets, place bets, sell bets, and many other things, including the benchmarking I talked about. Because when the agent is created the very first time, it's good to know at least somehow how good or bad it will be before it will lose the real money. So there is benchmarking that can be used to evaluate the performance. And lastly, the agent itself. So we have another open source repository with agents implementation, so you can run it by yourself, or at least check it out, improve, and review run it. That's totally fine. And also a few web page powered by Streamlit demos, where you can play with the agents itself. So not placing bets, but you can go there and ask a question and see how the agent is reasoning about it. And lastly, we have also this Jung dashboard, where all the agents that are currently placing bets on Omen are listed, and you can check out the accuracy, how much money they bet on, how much they won, how much they lose, what types of markets they are betting on, what questions, what are the answers, really anything. Any agent that will be on Omen should be eventually there to check it out. Yeah, and that is from my side. So, yeah, thank you.
Thank you so much. Okay, cool. Oh, please send our love back to everyone at Gnosis. Send our love to everyone at Gnosis. Okay, guys, we've got a break. We're going to break until 11:35. Go have a coffee, a little snack, and I'll see you in a bit. Everyone, we're going to start again. Do we have Sebastian somewhere? Okay, cool, guys. So we're getting started with our last session of the day, our last session of the conference. It's been lovely having you here. It's been so nice. I think we've all learned so much. I also just kind of wanted to touch back on like our intention with this conference, which was kind of looking at the issues in translational science and then kind of mapping out like all the ways DeSci could plug in and make things better. And I do really feel like we've kind of achieved that. And I hope you guys kind of are able to use what we've learned over the past 2 days as like a starting point and an inspiration point. Can we all sit down and just get ready to go? Yeah, it's like, shh. Okay, cool. So we have Sebastian here. Sebastian is a familiar face in the DeSci ecosystem. He's an investor in Molecule. He's a big part of BioDAO. I think he also sits on Molecule's board of directors. No, I lied. I take that back. He also, over the last 5 years, has co-founded 4 longevity biotech companies with a total equity value of over $6 million. So, that was entirely my fault. Sorry, guys. 600 million. Thanks for coming up. Yeah, here it is.
Yeah. All right. This is a beautiful weather day in Berlin. I'm happy to be here, and I was asked to start us off with my Donald Trump impression. Nobody loves Berlin like the Donald, believe me. In fact, I have a plan to make Berlin great again. We're going to rebuild that wall. All right. With that out of the way, I'll tell you about the DeSci. Oh, my slides are not showing. Is it possible to show them? Okay, cool. All right, so I'll tell you about the DeSci revolution, venture capital, and longevity biotech. So I'll tell you about my background, the technological stagnation hypothesis, some problems in the biomedical industry, glimmers of solutions, the DeSci vision, and give a shout out to LongBio, Prospera, and Vitalia. And if you like this, you can check out some of my other talks on YouTube for more details. So who am I? Sebastian Brunemeier. I look myself in the mirror every day and ask this question with tears in my eyes. But for the purpose of this talk, I'm a longevity scientist by training. I've been in the field over 10 years. I co-founded some prominent longevity companies and helped launch some venture funds. Today, I help run HealthSpan Capital, as well as ImmuneAge Bio, working on rejuvenating the immune system, targeting your bone marrow. Here are some other organizations you should check out if you're interested in, well, you guys know Molecule, but if you're interested in the longevity space. HealthSpan Capital. So I'm proud to say that we are the most active investors in the longevity biotech field. We've done about 30 deals in the last 2 years, and hopefully we will continue that pace. So here you can see some of the beautiful faces of the team, my partners. They've been at all of these prestigious institutions, et cetera. If you want to learn more, check out our website, healthspancapital.vc. Here are some of our portfolio companies. You can see them all on the website. And if you want to invest with us, you can invest through our angel list vehicle, and it's a minimum check size of 5K. So there you go, if you want to check it out online. Okay, so I think DeSci is the most important revolution yet. It liberates capital and technology from control of the corporation state. So this ties in with the technological stagnation hypothesis. So there's a great essay you should check out on the Founders Fund website. It's called What Happened to the Future? And it's summarized by Peter Thiel's quip. We were promised flying cars and we got 140 characters. There's been an advancement in a narrow cone of progress of software, but not in the deep tech world of atoms. And so you can read that essay at your leisure. Here's another interesting podcast with the author J. Storrs Hall on the Stranded Technologies podcast. It's my favorite podcast in the world by my friend Nicholas Anzinger. And he basically points out many of the ways in which bureaucracy and government actually stall innovation. We did an interview on the DeSci podcast with Niklas Anzinger. You guys should all check it out. Okay, so why is it a problem to rely on the state to fund science? Why is it a problem to rely on the government to fund science? First of all, it's a monopoly. Monopolies tend to not do a good job at anything. But one of the other reasons is that within academia, there's this phenomenon of crowding out. So there are only so many smart people in the world that can really advance science, and if you stick them all in academic ivory towers, they kind of fight amongst each other, they give grants to each other to work on trivial things, and it doesn't really result in that much progress. And so you can check out the work of the author Terence Keeley for more on why we should separate state from science, just like we separated church from state. Here's another podcast with Terence on the Bitcoin Standard podcast. So there's a decline in confidence in institutions, so the media, the government, the military, et cetera. And this is kind of to the credit of the public because they're starting to learn history a bit. Mark Andreessen gave a shout out for this book, The Revolt of the Public, where he said, all over the world, elite institutions from governments to media to academia are losing their authority, a monopoly, control of information to the broader public. This book has been my #1 handout to anyone seeking to understand the unfolding shift in power from hierarchies to networks in the age of the internet. And this is actually published by Stripe Press. They've got a lot of good banned books on there. So another problem with state-funded science is this enforcement of dogma, this groupthink. And we'll get into that a bit later, but we saw some kind of crazy dogmas in the COVID era, like it was once verboten to say, maybe COVID escaped from the Wuhan lab. And now, even the US government is acknowledging that that was the likely outcome. So progress has slowed in other domains too, like travel speed. You can see here, we actually flew faster with the Concorde, and we undid that. Also, we were producing a lot more energy until around the 1970s when we fell off the Henry Adams curve and basically banned nuclear power. So progress has stalled in a lot of different domains, but the one that I'm most concerned with as a biotech guy is the cost and time to approve a drug. That's getting exponentially more expensive. So friend of DeSci, Jack Scannell, has pointed out that this has been occurring for a while, and he called it E-Room's law, and it's a widely used term throughout the industry, that it's getting exponentially more expensive and timely to get a new drug approved. Also, Big Pharma is burning their investors' capital. So on internal R&D, they're actually burning the capital because their rate of return is less than their cost of capital. So they're really inefficient at getting new drugs approved when they're doing the research themselves. But that's why they acquire external research, which creates a great opportunity for people like us in the startup world. So I'll briefly, gently critique academia, a place where I spent many years. So a lot of it is like LARPing, live action role play. You get papers published without real impact. It's a lot like the Catholic Church. You have these grandiose buildings, you have people with these capes and these weird outfits, and then you can see Nature, Science, and Cell, the journals, the sacred texts that they pray to. So it's not really safe for truth seekers anymore. People get canceled a lot for not subscribing to the dogma, and so I think we can do better. It also pushes out really radical thinkers who advance the field. So someone like Richard Feynman was a little bit of a wacky guy. He would not have survived long in the academia of today. So we want to allow faster, easier access to funding for the renegade academics and unpopular hypotheses. Every breakthrough is unpopular at first. Nobody believes it. There was this one case of a guy who got a Nobel Prize for proving that bacteria, H. pylori, can cause ulcers. Until he came around, everyone thought it was just stress. And to prove it, nobody believed him. He had to drink a solution of this H. pylori and give himself ulcers. He's an Australian. So they're a little wacky. Okay, so here's a case study of how this dogma can really be destructive, and it's the amyloid hypothesis of Alzheimer's disease. So there have been very few drugs approved for Alzheimer's in the last decades. There are some reasons. One, the central paradigm of what causes Alzheimer's is probably incorrect, and we'll get into that. So most drugs in Alzheimer's have failed, and it's because of this Alzheimer's hypothesis that was enforced as a dogma by this cabal of scientists. And there's a great article unpacking it in Stat News, the maddening saga of how an Alzheimer's cabal thwarted progress toward a cure for decades. So long story short, amyloid beta is present in a lot of Alzheimer's brains, but it may not be the causal upstream factor. That's like blaming the firefighters for the fire. And actually, there were some amyloid beta targeting drugs that were approved recently, but it turns out they don't work. And there was this probe by various agencies into the coziness, the closeness of corruption between the pharmaceutical sponsors of the drugs and the FDA. And actually, the commissioner of the FDA herself asked for that inquiry. Okay, so how might DeSci help? So you guys know DeSci. A definition that I generally use is DeSci is an open source movement to source, evaluate, fund, execute, and commercialize new research without reliance on governments, large corporations, or established institutions. While financial profit is often sought, money is not the primary mission. DeSci aims to restructure the legal and financial foundations of science commercialization. So you can see some of these organizations, many of you are members of, our bio.xyz comrades have started a bunch of these BioDAOs recently. BioDAO is the one that's closest to my heart that I've been working with for the longest. And I'll get into longevity now. So big pharma is historically asleep at the wheel. So according to Jürgen Drews, the head of R&D at Roche, which is one of the most sophisticated big pharmas in the world in Switzerland, he said, biotech is taking up what pharma is neglecting, attacking the root causes of disease. And I would argue that there's nothing more at the root cause of disease than aging itself. David Baltimore, the president of Caltech and a Nobel laureate, said regarding the biotech revolution in the 90s, pharma was asleep at the wheel, unable to understand the profound opportunities of molecular biology and unable to take advantage of them. And that gave rise to companies like Genentech, one of the best performing investments in all of biotech. So let's get into longevity as the apotheosis of medicine. So some bad news is we're all aging and it's an exponential function. So even if we cured cancer in the whole population tomorrow, every type of cancer, on a population level, we would only get 3 to 5 years of extended healthy lifespan before something else would kill us. So to resolve the problems that are faced by government budgets and global demographic aging, we need to target the root cause, aging itself. And I would also submit that demographic aging is the most important problem facing the world. It's not climate change, it's not Russia or China or whatever. It's actually aging and disease which causes the most suffering, and it's 100% guaranteed. So the goal is to extend the period of healthy life by slowing the aging process. This is called health span. We can do this in animals pretty effectively. We can extend their lifespan over 50% in the lab because we understand the underlying molecular biology of aging pretty well, actually. Here's some of the mechanisms behind it. And we know there's out of date, probably 50 to 100 interventions that extend lifespan in mice and many other species. So part of my job is to translate this research in the lab into the clinic. Some of the favorite interventions and their quantum of lifespan extension shown here. I'm very partial to heterochronic bone marrow transplant, young bone marrow into an old person. Here's an example of one of these drugs, one of these interventions. So you can see on the left, you've got a mouse that has not received this longevity drug, and on the right, a mouse that is its litter mate, its brother, that has received that drug. And we know of many drugs that do this. Here are some of the smart cookies who are in the space, the social proof slide. Teal Bology is a big one, Vitalik, you guys know a lot of these names. So we have pretty clever people backing us now. There's exponential growth in the field, companies calling themselves long bio companies, even though a lot of these companies don't qualify by our strict criteria. If you want to know more, you go to longevitylist.com, run by my colleague Nathan Chang, that keeps track of all of the companies and job postings in the space. Here are some of the investors and company builders in the space. This is not a complete list, they're growing. Here's some of the companies publicly listed and privately held that are in the space. This is also incomplete. Go to longevitylist.com for more. Also, you can join a political movement called Vitalism. I just came from a conference in Valencia about vitalism, and I often say that there is a religion-shaped hole in the human psyche that people try to fill with something, you know, a sense of purpose and meaning and community. And in a secularizing world with less religion, we're actually filling it with bad stuff. So we're filling it with consumerism or nationalism or whatever. So let's actually give people something healthy to put in that hole in their brain, and I put forward vitalism. Okay, so some notes on medical freedom here. So if you only read a couple books this year, these are the books that you absolutely must read, believe me. They're incredible, tremendous books, tremendous. You've got The Network State by Biology, Sovereign Individual, which Peter Thiel wrote the foreword to, and The Art of the Deal. So, startup societies. This is probably not new to most of you, but you should check out Nicholas Anzinger's list of these new institutions, new ways to govern ourselves without relying on states. And so why is it urgent that we need to escape from government control? Governments are becoming more authoritarian, central bank digital currencies, taxes, inflation, lockdown, etc. You can try to cast a little piece of paper and hope that they listen to you, or you can vote with your feet and go to greener pastures, almost like escaping or defecting the Soviet Union. So people with means are moving right now. They're going to a certain type of place, and these are places that have political stability, low taxes, good weather, etc. And we should probably choose a certain number of these places to rally our forces, and then eventually we can have political influence in some of those locations. There's a very strong correlation between economic freedom and personal freedom and prosperity. Okay, so a brief word on FDA. I have a whole presentation on this, but we'll save that for another day. I'll just suffice to say, TJ, my favorite founding father, said, was the government to prescribe to us our medicine and diet, our bodies would be in such keeping as our souls are now. Wow. And in America, we spend like 4 times more than the closest country on healthcare, and we're like 40th in the rankings, and actually our life expectancy is declining. So something is really wrong with the system. We can get to that later. So I'll give a shout out to Prospera. Prospera is one of the most advanced, or the most advanced, startup cities in the world in the Caribbean. We did some events down there that were pretty successful. You can see it's this beautiful location on a tropical island. Roatan is just off the coast of Honduras. It's pretty easy to access. It's a major scuba diving location, actually. It's the largest reef other than the Great Barrier Reef in Australia. So we're doing various advanced therapies down there already. You can see some of the shots, some new buildings being built. Here's the master plan for this futuristic city. We have a regenerative medicine clinic down there that's been operating for many years. It's run by Glenn Terry, who's a very prominent orthopedic surgeon. He was on the board of the Olympics Medical Committee, so the sports event. He was one of the chief doctors there. Okay, so throughout history, we've had a bunch of different forms of governance, colonies, autocracy, democracy, etc. And there's a new one, which is sovereign individuals or something like that. And they're going to places like Prospera or Dubai or Singapore to get more freedom, economic and otherwise. And you can see the evolution of various forms of government, and they all have one thing in common, which is less state power. It is protecting the individual from the state, from the collective. And the state, the government, is not us. We are not the state. We are not the government. The state is a monopoly on violence of men with guns who try to convince everybody that we are them. Anyway, enough for my anarchist lecturing. So the dawn of the free private cities movement is equivalent to the US Constitution, a model the world will copy, causing a revolution in human freedom and flourishing. Check out this conference, Liberty in Our Lifetime, this winter. Coming up. It's got a lot of cool people talking about this kind of stuff. Another thing I'll give a shout out to, which is completely random, well, not completely. The first country that clones geniuses is going to take over the world within a generation or two, because intelligence is highly heritable. And imagine if you had 1,000 Einsteins or von Neumanns and they were working together, or you took the Manhattan Project scientists and cloned them, that country would take over. And I've spoken with some US government folks who actually see this as an issue of national security because the Chinese have been the first to clone primates and they have a very sophisticated cognitive genomics program. All right, so to conclude the visions for DeSci, you've got these layers of DeSci. Shout out to Niklas for this format. Some of the dreams for the future. So fully automated luxury cloud labs. As a scientist who's been pipetting late nights, it's just a waste of time and energy. So we can automate a lot more. We can make it seamless to launch companies. Good ideas get funded regardless of dogma, etc. So DeSci brings the computer revolution to the world of atoms, just as the printing press did during the Protestant Reformation. And we can disintermediate science. We can take a little bit of the influence of government, the state, and the big corporations, and the groupthink and dogma out of the program. Okay, so, and it's too late to put the cat back in the bag here. So now that this idea is out here, you can't kill an idea like this, especially with all these crypto people behind it. So with that, thank you very much for your attention.
Thank you so much. I always find your talks incredibly rousing. So yeah, maybe we can all start a molecule city. That would be so cute. Anyway, sorry, I digress. Next up, we have Mark. Mark has a degree in material science and mechanical engineering. He's currently working on the interface between humans and technology, building a brain-computer interface earbuds. He also is a DAO architect and works very closely with DAO and the Foresight Institute. So here he is.
Thank you very, very much. Yeah, so I've kind of been dabbling in DeSci for about 2 years now. And with this talk, I kind of wanted to give a background of building DAOs, things that work, things that don't work. A lot of people are interested in building their own DAOs, so I've been really enamored with the idea of creating DAO roadmaps to really understand how to build these organizations most easily and effectively. I also work as a brain-computer interface designer at Eden Technologies, where we develop brain-sensing earbuds. Alongside that, I was working at CureDAO, which is really more of a DAO which was building a product to collect a lot of data from people, also medical health records. And also from the Foresight Institute, I got a lot of inspiration over the past couple of years, also how I kind of got into the whole space, first working with technology trees to understand that we can create a lot of knowledge and understand what direction we need to go in, but then we need to build it. Also with like NeurotechX, I work a lot with doing hackathons, bringing people together, also just sharing general knowledge about neuroscience. So I try to put myself where I think it'd be most effective to build solutions essentially for the world. But I have a lot of inspiration also from obviously VitaDAO, the OG of DeSci, AthenaDAO. I also like to buy coins, so Genomes DAO, which I really love, Data Lake. So if I'm talking positively about things, I might have a financial incentive just to be fully transparent. But I really love to now talk about how all these learnings are coming into Cerebrum DAO to really take the best of what we've learned from corporate research, startups, academia, and bring it into something that we can build to enhance brain health and understand how we can help solve neurodegeneration. So Cerebrum is one of the, I believe, the youngest BioDAOs currently in the room. And we were inspired by Brian a couple of years ago. It's been building up now. We just launched our Neuron token. And that is at the point, I think, where you have some success, and then you say, okay, but we really need to build, release, and produce something valuable for the world. And this is really the crux where I think a lot of DAOs come into the fray. So I would like to first point out that science is not broken. I see a lot of talks where people kick off with that high point. Science is broken, we're here to fix it. I don't think it's fundamentally broken, but I do believe that with our new technologies, we can enhance the way that we're doing science. Because the scientific process, whether it's done by humans or DeSci agents or bacteria or any other type of autonomous agent, whether it's human or programmed, it's essentially flowing in the same direction. We have questions, we create hypotheses, we test those hypotheses with experiments, we create results, and then we communicate, we translate them into the world. How we translate them is the question. Is it going to be a paper that few people read? All right, I have a few research papers for my PhD. I probably have 300 reads since 2008. Probably didn't really actually have that big of an impact on the world, despite all of the Swiss taxpayer dollars that went into supporting my PhD. Do we ask the right questions at the right point in time? Do we put resources to solve those problems, those questions, at the right time? That's really where I think the DAOs are here to provide some value and solutions to the world. And to make that possible, I mean, a DAO is really like any other organization. You have energy going into doing something, and then you try to solve some of the easiest to solve problems first, and then you start to build value on top of that. So if we look at DAOs throughout the room or in the rest of the world, we see a lot of DAOs focused on trying to solve the funding problem, or the publishing problem, the collecting data problem, the understanding of how to develop and send resources, whether it's to automate wet lab work or to enhance the way that we create understanding from different publications, just so we actually know what we're actually creating in the world. And then obviously the IP fractionalization has evolved as a way to help to, let's say, bring a mechanism into play which allows DAOs to fund the research in a more fluid and scalable manner. And when I came into CureDAO a couple of years ago, I thought that there was kind of a roadmap. People like, okay, there's decentralized science. I learned about DAOs. I joined one. I thought, okay, we know how to do this. Someone knows how to do this. I don't think anyone really knows how to DeSci yet. That's why we're all in the same room talking about it. Also important to recognize that fact, because then we can be honest and share knowledge about how to learn how to do it properly. That's pretty much the thing I think about like continuously, I don't know, for at least a year, whenever I have time. Whenever I want to get a chocolate cake, drink some coffee, I jot down notes about what I've learned about different DAOs, how I think they're solving problems, how I think we should be solving problems for different focus areas, for example, neurodegeneration. So this talk is really about helping to translate a bit of my knowledge to the rest of the community. So how to DeSci? I would say defining purpose, rallying community, and then actually implementing and building is probably the easiest way to describe how to DeSci. That's actually not very fundamentally different than any other startup company or research project. We're all allocating resources towards our personal goals. How to do that with DeSci, given its unique challenges, is really one of the questions. So if we look at different DAOs, a lot of us are going through the same life cycle, same roadmap. It really depends, of course, on the purpose of the DAO. But defining the purpose is the first point. Not always the first thing that people think about. The first thing you might think about is launching a token, seeing a token go from 0.0035 cents to 80 cents over the course of a couple of months, and see your crypto wallet explode much larger than your bank account. Personal experience of mine. It's come back down since then, but I'm still happy that I joined that auction. Launching the token, and then solving the funding problem where we want to allocate resources to the right place is really one of the easy focus areas. True anecdote, when we were talking about CureDAO, we always use VitaDAO as an example, and we said, okay, this is really cool. We want to join this movement or enhance what we can do with data collection. But it was really easy because all they had to do is fractionalize some IP and launch a token. And we were talking about how to build up architectures, build up how to collect electronic health records. There are different barriers depending on what you want to do with a DAO. So if you don't understand from the beginning what you want to do, it's going to be difficult to build what's going to really have impact in the world. And we're very quickly moving to the point where we need to produce solutions, whether that is a solution pipeline within the DAO, whether it's bringing together resources and different products to enhance what the DAO is doing for the DAO participants. And hopefully, in my lifetime, I hope to see a point where DAOs actually start to dissolve because they're no longer needed because you've solved all the amazing problems that you set out to solve. And when we're thinking about timelines, I'm thinking about timelines going out decades easily, right? If you're starting a company, a startup company, you might have an outlook for 5 years, right? You launch, you get customers, you get acquired, you get sold. Hopefully you get a return to the investors in 5 years. I would hope that we are still around in 10 years, 20 years. If DAO is still going when I die, I think I would die pretty happy. And how we build out our plan for getting there is really something you should be thinking about early on. Doing the token launch, much like starting a company, is actually relatively easy, especially nowadays, right? I'm sure if someone wants to launch a new DAO within the next 2 hours, it's probably possible to do. Also with the smart contracts and the token launch, setting up your Discord, right? It's not mechanically very difficult. Getting people together, engaging the community for a week, a month, 2 months, a year, 2 years, 3 years, that's where the real challenges are coming into play now. And this is where you need to be building out those products that are going to facilitate that. So, I do love to play around with diagrams and roadmaps. I just naturally love to systematically take my background in design and product design and then to apply it to everything. So for me, thinking about a system of a DAO was the most natural thing when I first started getting into this field, you could say. And as a product designer, we use a lot of different non-technical tools, whether it's a design canvas, user journey mapping, talking to users, pulling in information, and I'd love to see more of that happening also in the way that we're designing DAOs, because really we're designing organizations for the DAO members. Like these are ultimately the people who are benefiting. And then beyond that, it's the rest of society. And the cool thing about thinking about this early on is that you can then start to track out your roadmap about what to build when, where you're thinking you're going to have the most impact, and then also understand what partners you need to work with to make this stuff happen, right? We know that we're going to create a lot of knowledge within DAOs, especially with the way that we're automating a lot of stuff. So we're going to need the AI agents, and we're going to need the large language tools and the coordination network to even make sense about what we're doing. I mean, even when I was working in automotive companies 20 years ago, I was blown away by how much we didn't really know about what was going on inside the organization. It's very natural to create more knowledge than you can consume. So these tools are filling that void that we all are going to have. So I'm really looking forward to having these open source solutions that are going to really transcend value across all the different DAOs. But that's not really helping us understand why we exist. Like, why are we here? So some of the tools I've been working with are the DAO canvas. This is basically just a natural extension of the business model canvas or the lean canvas, right? Every time there's a new cool thing coming up in the technology world, someone now builds a canvas for it because it's very, very easy to create a two-dimensional map which shows where you're going and what resources you need to allocate to bring together to make it happen. So the DAO canvas is one thing we created, which includes key points like tokenomics, growth path, deal flow. Again, really trying to tune this for DeSci projects. But if you want to get more complicated, more multi-dimensional, you can start looking at a strategy canvas. Strategy canvas, it's really cool in my opinion because it's not super popular yet, but it combines a lot of different metrics and dimensions also within a 2D space. So you start with your subject in the middle, you break out your vision, and then you start to break out, essentially, you can basically trace a path from one side to the other, and that can then be transferred over into your roadmap. So if we do that for DAO, we understand that we want to essentially enhance brain health. We want to solve neurodegeneration issues right at the center. If we start to break it down, we can zoom in, we can see that for our mission and vision, it's going to be about defining why we exist within the actual solution landscape that we currently have. Because the DAO is just one type of organization, right? There's other companies that are addressing brain health, there's pharmacological interventions that are coming out, and our question is, how are we making a difference? Is it just going to be about funding projects that might have a payoff in 3 years or 5 years or 10 years? Well, that's really cool. And that's one way to create a DAO. But what's going to happen in the intermediate years? What's going to happen next month, in the next quarter? What are the DAO members in Discord going to say to us when we say, okay, we're funding these projects, and that came out of our treasury. Okay, now our treasury is kind of gone. Do we have to sell more tokens? Do we have to raise another token round to have more money to do more investments? No. What we can do is create products which are impacting brain health of the DAO members today, tomorrow, over the next couple of months. And the easiest way to break down the problems that you have for a DAO is to say, if X exists, then Y is possible, and Z will happen. I actually learned this over in Gorlice Park when I was working at EF on an entrepreneur summer vacation. If you can break down your DAO concept into a few simple sentences, you can communicate it to other people, and you can understand it better for yourself. Because every DAO is going to get super complex in the world, right? More data, more DeSci agents, more experiments. It's never going to get easier to filter down to a simple question that you want to solve than when you start it. And with neuroscience and neurodegeneration, it's relatively easy, right? If we can understand when amyloid plaques are developing, if we can have early interventions, then we can start to bring in new ways to treat the disease. We can open up new ways to understand how we're aging over time. So for the implementation of DAOs, much like products, I like to go through from 0 to 1, from idea to solution. And it's really important to implement, I think, in a more traditional product sense, going from trend mapping, user stories, defining the key features you need in your DAO, the key products you're going to need to bring out to your DAO members, which is really going to bring value to their lives. If you've never been in a team before, or if you have been in a team before, I'm sure you've gone through the forming, storming, norming, performing concept. It's coming back from the 1950s, and it's really important to understand and give expectations to new DAO members that a DAO is not a magical thing which just makes everything easier. You're going to go through a lot of friction, a lot of hardship, and probably a lot of emotional roller coasters when things work, they don't work, the token price is up, the token price is down. But getting through these hurdles is what is going to give you sustainability over the next decades. In that, roles and responsibilities, easily, I think, one of the most important takeaways. Someone has to be in charge, either a core group or a core number of stewards in the beginning, because this is going to help define the whole culture for the DAO going forward. Culture management, what problem you're solving, how you're solving it. This is stuff that everyone has to understand and really agree upon and be aligned on. Because if you're not aligned, you're not going to be building and engaging the community in the same way. Like, for example, right now with Cerebrum, we're first engaging with the deal flow process. Right, a lot of people can come in, they can be reviewers, they can be deal sourcers, and they can really be part of creating that funding pipeline. But going beyond that, going beyond assessing deal risk or deal impact, which is really cool, but also like a very minute, interesting, and sometimes frustrating part of the whole process, we're looking at how we're going to create circular engagement. So one part is going to be bringing out applications which DAO users can use to assess their brain health, to assess their neurological, their brain health journey going forward. And if we now connect that with tokenomics, with the metrics of the neuron token, and the ability to collect data and use it for understanding interventions, generating knowledge, and then ultimately coming back and generating value, because users will understand then which interventions, which supplements are actually having a positive impact on their brain health and their life. That is going to give us the road forward to really have that multi-year engagement and retention of DAO users. We also have a lot of work going on within product development and architecture for larger data products, which is really going to build upon that circular notion. And I believe it will give us the ability to go to that 10-year, 20-year, 30-year, hopefully 100-year, and it'll still be there when I die. So launch, build, release, bring value to the users, to the DAO, to the world. That is the most important thing I would leave you with today. Happy to have you in Cerebrum DAO. Join our Discord. Hit me up on Twitter. Happy to talk about this anytime. Thank you.
Thank you so much. I mean, one of the things I've always thought is that these sort of maps that you make will exist also to benchmark us as we grow, you know, how a DAO runs in this year versus 2 years versus 5 years. And I think the work that you're doing is going to be very useful for tracking our progress. Okay, next up, we have something a little bit different from what we've been doing. We are having a pitch contest. So 4 sort of companies that have gone through the Nucleate activator program are going to pitch for you. You guys are going to vote and they're going to win a prize, so it's very exciting. Logan's going to introduce them, and then Suyen, who you met yesterday as well, who's been one of our Nucleate fellows, she's going to be the glue that sticks it all together. So yeah, very excited. Let's go.
Thank you, Ella. So, Friday afternoon, you guys have been paying attention for a day and a half now, and all I'm asking for you is another 20, 30 minutes of your full attention because you're playing a very important role today. So I'm joined here by Suyen, who you met yesterday. She is the director of the Nucleate Activator program, and together they help kind of incubate and form and spin out teams from the program, and 4 of them are here today. And they are here to win your hearts and minds in a pitch competition. So the 4 teams have a 5-minute pitch, and there's going to be a QR code up for voting afterwards. We're going to hold the voting open for about 2 minutes, so everybody in person and everybody online, please vote, let your voices be heard. Very democratic, very DAO-like, you know how we do it around here. And they are competing for some prizes, so some cash and kind of support prizes from Molecule as well. So I'm going to let Suyen take it from here, introduce you to the teams, and everybody pay attention and get ready to vote.
Hello, everyone. Thank you for staying all this through DeSci.Berlin. It's so good to have you here. Talk to so many amazing people. Again, this is my first DeSci conference, amazing. But right now, I'm here to showcase 4 out of our 8 companies that have gone through the activator program this past 6 to 7 months. I'm happy to have them all here. And because it's a project, it's the first activator program that we do in Germany. This is our baby. We have walked through this road with all the companies. So the first one is Alexander Sontag, and he's a doctoral candidate where he focused on gene therapy and molecular biology, and today he's going to be showcasing their company, Escape Agent.
Thank you. Okay. Perfect. Yes. Hi, we are Escape Agent, and we are tackling one of the most urgent problems faced by the gene therapy community. Through the power of genes, all processes in living being are regulated. Gene therapy taps exactly into this potential to treat a wide spread of diseases and to improve the quality of life. Today, we are able to produce gene therapeutics that survive the path from the syringe to the cell. However, once arrived, a second line of defense is waiting and trapping the genetic material. It's industry-wide acknowledged that only 1% of the genetic material reaches the site of action. This is so far overcome by using high dosage, which in contrast leads to higher costs and more side effects. Our escape agent is a molecule that interferes exactly in this process. It helps the gene therapeutic to escape the defense mechanism or the so-called endosomes. By doing this, more genetic material is reaching the site of action, which allows lower doses. We could already demonstrate this powerful delivery enhancement in vitro in over 10 cell lines and have encouraging in vivo data. While normal gene delivery is quite inefficient, the simple addition of escape agent leads to a dose-dependent 20-fold improvement, even at high concentration where structurally similar compounds are toxic. The great potential of escape agent could not only be proven in our lab, but was also independently confirmed by several academic and pharma company partners. As an enabling technology, we are joining the ranks of delivery tools like adeno-associated viruses and lipid nanoparticles, but are more related to endosomal escape triggering technologies like cell-penetrating peptides or other distinct small molecules. However, escape agent stands out to its great efficiency enhancement, its good therapeutic window, and its defined analytics, making it a prime candidate as an additive for gene therapy. Through co-development of an exemplary gene therapeutic drug with a pharma company, we will establish a proof of concept in human. At the same time, we will work towards a drug master file, which facilitates new investigational drug application, which, with the aim to provide our technology to a wide spread of companies. Therefore, we will have all regulatory aspects cleared by 2026. In addition to further development of our technology, we plan to establish a high-quality GMP production route and strive towards several clinical trials incorporating escape agent. Here our fantastic team is key. I'm very happy to work with Diana Munte, an expert in regulatory and GXP, and Dr. Jan Niklas Dürrich, who as project manager is the connection to our experts and our cooperation partners. I, Alexander Sonntag, will further develop the technology and our product pipeline. Our biggest gratitude goes to Nucleate and our expert for their special support. Together, we will empower the gene therapy industry to pave the way for affordable gene therapeutics with less side effects. Thank you very much for your attention.
Thank you very much, Escape Agent. If you are more interested in the technology or want to learn more about this, maybe one of the DAOs is also interested, approach later to the teams. We won't have time for questions. This is going to be a speed pitch competition. But next is one of our, so Nucleate has 2 tracks, a bio and an eco track. And next up is Fabian. So Fabian is the co-founder and CEO of Fluvion. He holds a master's degree in international management and has a lot of experience in the pharmaceutical industry. And today he's going to be presenting about what Fluvion is trying to do for the environment.
Sia, can I have the presenter? Okay, perfect. So, hi everyone, I'm Fabian. As you already heard, co-founder and CEO of Fluvion. I think most people here in the room will consume biological liquids on an almost daily basis. The most important one, or most prominent one, is milk, but there are also others, like for example silk, the musk in your perfume, or certain pharmaceuticals. The problem with these is that currently they're produced via conventional agriculture, so with animal usage. Brings a lot of ethical complications, but also issues for climate. 1 billion tons of CO2 annually just for milk, but also limits the availability of especially the pharmaceutical resources quite severely. So actually it would be time for synthesization. However, with the current methods, each individual molecule requires an own process with its own bioreactor, making the whole thing very capital intensive and very ineffective. So we thought, well, this can't be the last word in this matter. So with Fluvion, we've been developing a novel type of bioreactor we call the biomimetic bioreactor that is able to produce these liquids in one go in the quality we know. So it's like 100% milk, 100% musk, no replacement. Apart from that, also the advantages are that it's like very easily scalable. So once you reach a certain threshold, you can produce high volumes very, very quickly. It's epithelial cell agnostic, so it works well for the milk, but you can also apply it for chemicals or pharma. And due to the reduced purification necessities downstream, you can also significantly reduce the production costs depending on the application. Currently we're like in what I would call the end of the first trimester. So we have the cell lines, they work, we have first promising results with the membranes, looking to reach MVP stage by end of or mid next year, then scaling up to mid-sized bioreactors to then reach, let's say, full-size industrial application sites by the end of this decade to be ready to tackle large volume markets. That's also why we're raising a pre-seed round of 500,000 right now. Main funds will go towards membrane research and cell research. We don't want to do this B2C, but to maximize our impact, we want to do it B2B, so that other companies, our corporate partners, can produce these high sustainably, high quality liquids in-house, optimize their processes in the meantime, and reduce their production costs. In return, that allows us to generate multiple recurring revenue streams around the sales of these bioreactors, specifically licenses, support contracts, as well as the cell cultures, capturing around 50% of the production costs at our in-house. In this niche of the liquids production, we differentiate ourselves mainly through the bioreactor itself, which has unprecedented level of scalability and associated production costs no other competitor can match so far, which is also confirmed by multiple expert interviews we conducted, and these see actually very high potential in the technology, but also the idea itself. Now, I talked about a small market. Actually, it's quite big because it covers multiple segments, 400 billion annually worldwide. So actually, more interesting is the direct addressable market, 8.6 million we estimate by the year 2030. We aim to capture around 200 million of that by the year 2032. Entering the market mainly through, or first, through the high-priced chemical complex aromas and venoms, because here's a quite good combination of regulatory and pricing structure. To then, which allows us to generate quite a promising and high growth business model. So last but not least, who's behind? You heard a bit about myself. I'm Fabian. I'm responsible for the business side, everything outside of the lab. Responsible for the research is Marcus, my co-founder, very experienced and expert in cell and molecular biology. He has a PhD from Cambridge, won multiple prestigious prizes, including from the National Academy of Sciences of Germany, the Leopoldina, and prestigious EU fellowships. We're supported by a bioprocessing specialist and a biotechnologist, which will be expanding continuously throughout the year. Additionally supported by a very motivated and capable mentoring board covering all aspects of early stage research and development, and are quite well connected to the pharmaceutical industries. So, if you have any questions, just catch me later. I'm always happy to talk. Thank you very much.
Thank you so much, Fabian. Next, we have Jellyfish Bio. And Jellyfish Bio is going to be showcased by Nikita. So Nikita is originally a computer scientist and have co-founded 3 IT startups in diverse fields like crypto and neuromarketing. So those crypto peeps in the crowd, come talk to Nikita afterwards. And here's Nikita to talk about Jellyfish Bio.
Hi, Andreas. Yeah, thank you. Quite a great event, and thank you for this event and Nucleate for this opportunity. So, I'm Nikita from Jellyfish Bio, an AI platform for genetic diseases blood biomarker discovery. So, why do we need such platform? First, existing approaches for genetic disease monitoring don't work. They mostly rely on complex but infrequent in-hospital examinations, and it fails in several scenarios. First, for rapid disease progression, we have insufficient number of patient visits to track progression. For slow disease progression, we have not enough power to detect minor changes in health status. And finally, we fail to detect incidents like, for example, liver failure, if they didn't happen in clinical settings, which they usually not. And finally, we cannot use advanced data analysis techniques like deep learning because in our case we have rare patients and small amount of visits to make this kind of analysis. At the same time, 80% of genetic diseases are multisystemic, meaning that we don't have one good outcome measure, like, for example, tumor size in cancer, and we really need progression biomarkers to track disease severity over time. But researchers in our field know that there is a huge lack of good progression biomarkers for genetic diseases. And it's crucial for clinical trials. Use of biomarkers in drug development process increase the likelihood of clinical trial success by 30%. So I'm Nikita, PhD in computational health, together with my co-founder Dima, who is PhD in medical genetics with focus on blood biomarker discovery, and awards from American and European Society of Human Genetics come out with Jellyfish Bio. So we focus on most universal way to detect biomarkers, blood biomarkers. We utilize different approaches like proteomics, targeted metabolomics, and lipidomics to find biomarkers. Next, we developed mobile application to collect wearable device data and patient-recorded outcomes to make monitoring of patients actually continuous. And finally, we use this deep annotation to create to find blood biomarkers even in small patient cohorts. We prove our approach in science, where my co-founder Dima found blood biomarker for mitochondrial disease patients, which together with phenotypic data overperform all existing biomarkers in our field. So our uniqueness in early stage collaboration with clinicians with access to rare patients. So we create some kind of community of clinicians who have access to rare patients. We provide them platform to make their researches on our platform and secure funding for their researches. Next, we create joint IP rights with them for new disease progression biomarkers as well as additional outcome measures for genetic diseases. And our current focus in two fields, in metabolic diseases, genetic disease, and neuromuscular diseases. So our business model, first we start with R&D with patient continuous monitoring in R&D settings, and all the way down we license our discovered biomarkers to pharma companies in all the phases of drug discovery till the drug development phase, till the developed drug phase, when we aim for revenue share from 2 to 5% with pharma companies. It represents 9 billion market opportunity on over than 300 billion market of genetic disease treatment. And we work on this project for 5 months, but we already started our first clinical study in Japan where we monitor 23 patients with Fabry disease. And in our pipeline, we have 4 more studies, including Parkinson's disease. So currently we are raising $500,000. Most of it will go to finalize product development and first licensing, like with Fabry disease, we aim to license first outcome measures for clinical trials till the end of this year. So if you want to know more about biomarker discovery and genetic disease, come talk to us. Thank you.
Amazing, thank you, Nikita. So we have our last pitch. Have your phones ready right after Anna's pitch. It's gonna be a QR code where you all get to vote, not just people here on site, but also everyone that's on the live stream. So last but not least, I am pleased to introduce Anna. And Anna holds a PhD in medical biotechnology, and it's currently affiliated with doing a postdoc at the Charité here in Berlin. And I have Anna now to showcase Silica corpora.
Thank you. Okay. Imagine you go fishing and you get the fish that you want. At Silica corpora, we are the skillful fisher, but instead of getting all the fishes, we get therapeutic antibodies. We can design holistic antibodies with generative AI. And with this approach, we are also unique because we can design functional antibodies only with the information of amino acid sequences of the target. And with this, we are 3D structure independent from the target and do not accumulate errors as our competitors and other AI companies do. At the moment, we have 4 different modules. the app mapper for epitope mapping on the target, the generator for de novo design of therapeutic antibodies in any formats, the discriminator, using it, for example, for saying discriminate between binary and non-binder, and the optimizer that can optimize antibodies for any characteristics. Our platform is validated. We ask our platform to design candidates against HER2. HER2 is the marker in breast cancer. And from 200 trillion, we got 600 candidates within days. And trastuzumab, the therapeutic antibody that is used in the clinic to treat breast cancer and to target HER2, was among all those candidates. And we took only 10 candidates from the 600 and tested them in the wet lab. And now I want to show you how results can look like. So here you can see ELISA results and the binding of the candidates. In red, you can see the binding of trastuzumab. And we took only 3 candidates that were designed from our generator and optimized them. So these are the mutants you can see here. And you can see 3 arrows indicating mutants that have even better binding than trastuzumab, what you can see here in red. So our platform is able to even design better candidates, therapeutic antibodies, that are actually currently used in the clinics. Our team consists of 3 different pillars. Tim, our CTO, who has more than 10 years experience in AI drug design. Jaime is our CEO. He has experience in project management in many different companies. And me, I'm the CSO and have a PhD in biotechnology. And of course, we also have famous key advisors that support us in different areas. So far, we already secured 300,000 in our pre-seed round. And we're part of Berkeley Skydeck and are part of Exist. Our platform is validated with external partners, and we already had our first paid customers as with the Charité. So we look for our tailored solutions, and we want to make fallen angels fly again. And we believe in long-term cooperations. And we are looking, or already looked for, co-development of therapeutics. In our seed round, we asked for 1.5 million euros to cover the next 18 months of runway. We want to test our own therapeutics in the wet lab, complete our IP strategy, and want to build our own foundation models for antibody design. So be part of next generation therapies, be innovative, be a skillful fisher. My name is Anna, and thank you for your attention.
Okay, amazing job to our 4 companies. Now we're gonna show the QR code. Have your phones ready, so everyone can get to vote. So people here, people on the live stream, I'm gonna do that myself too. Waiting time, drum rolls. Yes, so people that are on the live stream, please also take out your phones, iPads, computers, however you can just take a picture and go to the type form and get your vote in. One more minute. So if all of the 4 companies can come up stage, please. The moment of truth. 30 seconds. So get your vote in. People also on the live stream. Yeah, come. Well, we wait for the few more seconds. I just want to say, so it has been amazing to be working with all of these guys. I was in the first round of interviews when we were just trying to get companies into the program, and it's amazing to see the development and where you guys are now from the first day where you were pitching me your company. It's amazing. I truly believe and I hope that the Nucleate Activator program has provided some mentorship, expertise to your companies, and most importantly, connections, networking. If anyone here is interested in talking to any of these amazing companies, please do so after we're done. And I think we have results.
All right, moment of truth. Can I get a drum roll from the audience, please? All right, the winner of the first DeSci.Berlin pitch competition is Silica Corpa! Woo! Would you like to say a few words about your victory?
Thank you so much. Actually, this was my third time on stage, so that's why I'm really excited to win. Yeah, thank you, Nucleate, for the support. It's really great, support, and I think I don't have any more to say. Thank you.
I do have one more thing to say. So these amazing 4 companies, along 4 more other, are going to be showcasing their companies at Demo Day. Demo Day so it's the final day of the Activator program where they get to show everything, what they have gone through the program, their companies, and we have a QR code for you to join. It's going to be here in Berlin. So everyone that's based here in Berlin, please come, even if you're not. Travel and come. It's going to be an amazing day. It's going to be a day not just for pitching, but also it's going to be panelists. It's aimed to be a full day of networking as well. People in biotech. I know some of our molecule people are going to be around there. So, but yeah, you can just scan the QR code and make sure that you are signed in for the event.
Okay, cool. Thank you so much, guys. You guys were all great. I honestly was so impressed with the quality that you guys had. Okay, with that, I'm going to close DeSci.Berlin 2024. Thank you so much for being such a wonderful audience. It's been spectacular. We're going to, like I said, have this sort of after party at a beach bar directly after this. And also just a reminder, we do have another event coming in after us quite soon. So I'm going to request that you guys sort of move with haste well, at least out of this venue. And I'll see you guys all at the beach bar. Cool. Thank you so much, guys.